The role of DNA when it comes to protein synthesis. The topic today is the role of DNA in protein synthesis. So what role exactly does the DNA play when it comes to the synthesis of proteins? And what exactly are these proteins that we're talking about? Okay? What other organelles are involved in this particular process of protein synthesis? That is exactly what we are going to look at today. We are going to look at the role of DNA, and also we are going to look at another form of a nucleic acid called an RNA. That is the outline of today's lesson or today's tutorial right here on Darasa with me, Mr. Ben. So all that I need you to do is just stay easy, give me your maximum concentration, take a writing pad and a pen, and then from there, let's take a walk together through this journey of the role of DNA in protein synthesis. Thank you. Before we look at the role of DNA in protein synthesis, let me give you a good background. Let me give you a good background of what you need to know before we get into the real stuff, the gist of the stuff. Okay? So here's what you need to know. What proteins are we talking about? Can you give me 2 examples of proteins that we're talking about here? Okay. Someone is saying meat. Someone is saying beans. Okay. Another. Okay. Those are proteins. Those are proteins, but they are not the type of proteins that we're talking about here. Okay. The type of proteins that we're talking about here, we are talking about number 1. We are talking about structural proteins. Okay. Let me give you an example of a structural protein. Look at the structure of a cell membrane. If you look keenly at the structure of a cell membrane, you will realize that we have a part within a cell membrane that we call a protein layer. We call it a protein layer. So that is a structural protein molecule. That is an example of a protein. Another example of a protein is a functional protein. A functional protein. And let me give you an example of a functional protein. An example of a functional protein is an enzyme. You know what an enzyme is? An enzyme is basically an organic catalyst that speeds up the rate of a metabolic reaction. Give me an example of an enzyme. I'll give you one. The first example of an enzyme that helps in the digestion process. Let's talk about ptyalin. Ptyalin is also called salivary amylase. So that is an example of an enzyme, and that enzyme is a protein molecule. Another example of of a protein, specifically a functional protein, is hormone. A hormone, or rather hormones. Hormones are functional proteins. Give me 2 examples of hormones. Any hormones you know? Maybe one plant hormone, one animal hormone. Let me give you one plant hormone. One example of a plant hormone is abscisic acid. Okay, those are examples of hormones. Okay, abscisic acid, gibberellic acid, auxins. Those are plant hormones. Okay, those are plant hormones. Each of them, they have specific roles. Now let's look at animal hormones. Any example of animal hormones? Okay, we've got these hormones, reproductive hormones. For instance, testosterone. Testosterone is a reproductive hormone in males. You know what it does? If you don't know, just stay put. In our lessons, when we'll be talking about reproduction, you'll get to know the role of testosterone. But right now, we are talking about the role of DNA in protein synthesis, and I've just given you the examples of proteins that we're talking about here. The examples of proteins that the DNA molecule is involved in synthesizing. Okay? So, having known that those are the examples of proteins that the DNA synthesizes, let's look at the structure of the DNA molecule. If you look at the structure of a DNA molecule is made up of 2 strands. Okay? DNA molecule is made up of 2 strands. A DNA molecule is just like a twisted ladder. Okay? A twisted ladder. You take a ladder, you know how a ladder looks like, and then you twist it like that. So that is how a DNA molecule looks like. So this one end is a strand and this other end is a strand, and then these staircases are bases. Okay, to be specific, let me call them base sequences. Let me call them base sequences or codes. C-O-D-E-S. Codes or base sequences. What are these codes? These codes are essentially base sequences. A base sequence is a sequence of nitrogenous bases, okay? A sequence of nitrogenous bases, the alignment of nitrogenous bases along a strand. The nitrogenous bases are for adenine, thymine, guanine, and cytosine. So the alignment of these nitrogenous bases along a strand, a DNA strand, that is what we call a base sequence, and that particular base sequence is a code, okay? And that particular code, it determines the particular amino acid that is going to be formed. Or let me put it differently, that specific codes within a strand of a DNA molecule will dictate the type of protein that will be formed, will dictate the type of amino acid that will be linked, in turn, which will form a protein molecule. An amino acid is basically a building unit of a protein molecule. Just like the building unit of a carbohydrate is glucose, okay, or a monosaccharide. So, the building unit of a protein molecule is an amino acid, okay. So, if you combine many amino acids together, you form a protein molecule. So, these amino acids will determine the type of protein that will be formed. And the type of amino acid that is linked will be determined or will be dictated by the code that is along a DNA strand. So that is important for you to know, okay? After that, let me introduce you when, for instance, let me give you this. When, for instance, you have 3 base sequences, or let me put it differently, you have a combination of 3 bases. You have a triplet base. Of course, you know triplet, tri means 3. You have a triplet base. That is, for instance, A. A denotes adenine. It represents adenine. So you've got adenine, 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 or thiamine, thiamine, thiamine. So a collection of a triplet base forms something that we call a codon. I repeat that again. A collection of 3 bases, forms a codon, okay? And this particular codon will determine the type of amino acid that will be linked to it, which in turn will form a specific type of a protein molecule. Let me tell you what that means. Let's take AAA, just what I've said right now. AAA, that is a codon on its own. So if you take that particular codon AAA, that codon, it will link, or it will be linked to a specific amino acid, and that specific amino acid is called lysine, okay? That specific amino acid will be called lysine. So lysine will only be formed where there is this particular codon called AAA. Where we have AAA, the only amino acid that will be linked to that particular codon is what? Lysine. Another example, when we have a codon, let's take T. T, remember, T stands for thiamine. So when you have a combination of 3 T's, that is 3 thiamine molecules, that will dictate the specific amino acid that will be formed there, or the specific amino acid that will be linked to that particular codon. And the specific amino acid that will be linked to that codon is phenylalanine, okay? Phenylalanine. So phenylalanine will only be attached or linked to this particular codon TTT. Lysine will only be linked to this particular codon called AAA. I hope that we are together up to there, but I don't mind repeating because these may sound a little bit intricate. If you don't follow closely, I may end up losing you. So let me go back a bit. So I'm saying that on a DNA strand attached onto a DNA strand, we have base sequence. We have a base sequence or base sequences. Base sequences is basically the order in which the nitrogenous bases are lined along a DNA strand. Okay, the order in which they are lined. Remember, the nitrogenous bases, they are like staircases on the ladder. Okay, so the base sequence is the order of nitrogenous bases.